mutations in the obese and fatty acid genes in rats, mice, and humans. Through this co-discovery of hormone leptin and cloning the leptin and leptin receptor genes has made a
Recording in
major
progress.
role in understanding human obesity. He has published over 300 peer-reviewed scientific papers so far, which has been cited over 13,000 times in the world scientific literature. He has received numerous awards for scientific and pioneer work in medical research, such as Presidential Medal by Obesity Science Society, Albert Ronald Award of the American Diabetes Association, Mentor of the Year Award in Columbia University, and so on. So today, he is going to give us a lecture on studies of human body weight regulation. I'm going to stop here and let's welcome Dr. LaBelle. Thank you.
Well, thank you very much. I don't often or frequently talk about this particular topic, which is one of great interest to me and in which I've done, along with collaborators, a fair amount of research. And I picked it for this evening, maybe this morning for you, in order to give you a sense of my sense of where the most important or some of the most important questions in metabolic physiology relating to human body weight are sort of currently confronting us. I think many of you, certainly I, work at a very focused level on questions having to do with various aspects, as you mentioned, of the genetics and the physiology and the neurobiology. But sometimes I think we lose track of what the sort of overarching issues in the field are when we look actually at how humans regulate body weight. Because in terms of understanding both the genetics and the physiology and certainly the pharmacology, as I'll show you, we really need to understand this very well in order to get focus on the areas that are most important to be able to both prevent and treat this disease, which only recently, as a result of these new GLP-1 receptor agonists, has become tractable from a pharmacologic point of view. But as I'm going to show you, the GLP-1 receptor agonists are not the final story or the final answer, because once those drugs are discontinued, the physiology that I'm going to talk to you about simply returns. So it's very important to understand this, at least I think it is. And I think new insights into this physiology will help in the management and ultimately the prevention of the disease. So let me give you a little sort of overview of my thoughts on this, what I think is a very important issue. Well, maybe not. There we go. Okay. So I want to just remind you of the fact that body weight in humans actually is regulated. I think if I made this argument to you about mice or rats or other organisms, you would probably be more likely to believe it. But what I'm going to show you actually is that human body weight is regulated, and that regulation has a critical role in terms of understanding what can and should be done to either prevent or treat the disease. But at a very high altitude, just from an evolutionary point of view, why would my assertion that body weight is regulated likely be true? And I think the argument is that body fat, when we talk about body weight regulation, we're talking really primarily about body fat, as you'll see. And body fat needs to be protected, not only in humans, of course, but in all mammals and other animals, in order, number one, to secure reproductive capacity. In a human, in a female whose body energy stores drop below about 100,000 kilocalories, primarily in the form of fat, hypoamenorrhea results. So women who have either anorexia nervosa or are very accomplished athletes who tend to have low body fat levels, many of them do not have full ovulatory function on a monthly basis. And it's because of the fact that they don't have enough body fat. As you know, or we now know, leptin, which we'll come to in a moment, plays a critical role in the assuring the integrity of the reproductive axis, the gonadal axis. And when fat is insufficient, leptin deficiency, which results from the loss of body fat, actually helps to shut down the gonadal axis so that pregnancies that can't be carried all the way to fruition and successful feeding of a fetus don't get undertaken. And the second reason that body fat needs to be regulated upward or defended, kept above a certain minimum level, is in order to be able to survive under environmental circumstances in which adequate food is not available. Now, this isn't a big problem today, as we all know, but certainly 100,000 years ago, it was a big problem for our genetic forebears who had to survive under circumstances in which adequate amounts of calories were available on an intermittent basis. And it was important to have a form of energy storage that would permit an animal to keep going despite not being able to secure meals on a regular basis. And body fat serves that purpose. Why would you want to make sure that body fat didn't go over some maximum level? In other words, why would you want to make sure it was kept below some maximum? And there, it's for the reasons having to do with the environmental vicissitudes. If you're hunting something, you need to be able to stay alive to be able to do it, but you don't want to get so overweight that you would fall out of a tree or be able to be trapped or caught by some other predator. So excessive body fat certainly is something that you'd want to avoid. But if you consider what our evolutionary history is, we've had a lot longer in the presence of inadequate or reduced or not fully sufficient access to energy intake or food intake, much more than the much more recent situation of having excess calories available. So you would predict that evolution would have designed genetics and physiology very much more strongly in the defense of against loss of body fat than against its gain. And even if you had never thought about this that way, if you look at what's going on in populations around the world, that's clearly what's happening. If there were fully effective defenses against gain, we wouldn't have the obesity epidemic that we do. But the reason that we do is that there is this very strong regulation to prevent body fat loss below some minimum level. And that, I think, is the critical issue with regard to human body weight regulation, as I'll show you. So in order to understand or have some perspective on what I'm going to talk to you about, you need to understand just on a very basic level the way that the first law of thermodynamics relates to actual biology. And the first law states in a physical sense that changes in stored energy or energy in the system is equal to the difference between the energy added in the form of heat and the work done by the system. So delta U, change in internal energy, is equal to the difference between what you might describe as the work done by this of the energy put into the system minus the work done by it. This is the first law of thermodynamics, and it relates absolutely to human and mammalian energy homeostasis as well. In the case of the biology we're talking about, the change in stored energy is what I've referred to here as the soma, the body, primarily in this case, adipose tissue. The energy added to the system, in this case, is energy intake, shown by EI, and the work done by the system is energy expenditure. This is it. I mean, this is basically the premise around which body weight and energy homeostasis are based. It's first law of thermodynamics applied to a biological system. So if you look at the energy expenditure of a human, this is the EE side of the equation right here, the EE. These are the compartments of energy expenditure in a sedentary human being. Resting energy expenditure accounts for somewhere around 60% of 24-hour energy expenditure. It's affected by all of these factors that are shown here. Diet-induced thermogenesis, which is the energy cost of digesting a meal and distributing the substrates into the respective tissues, accounts for maybe 5 or 7% of energy expenditure. And the rest here, this remaining 25 or 30%, is physical activity, which is the energy cost, of moving around at various levels of activity. Obviously, if you're very physically active, this percentage will go up. If you're totally inactive, it'll be even a smaller fraction of this 24-hour energy expenditure. But these are the three major compartments, and we'll come back to this in a moment. So consider the following. This we sometimes refer to as the truck of life or fundamentals of nutrition. During a lifetime, a human being actually takes in this amount, this obviously is a rough estimate, about 28, 40, or 3 tons of fat, 3 tons of protein, 8 tons of carbohydrate. This is in roughly a 70-kilogram man. So we're talking about tons of food and water that go into this individual, who himself is comprised of very small amounts of these materials. The majority of the weight of a human is in the water component, actually. But what I'm trying to do is show you that massive amounts of food go through a human being over a lifetime and are basically spent, that's the energy output side of the first law, as heat, physical work, and then there's gas exchange that releases some of the substrate as carbon dioxide and fecal waste. So the question is, what's the balance here? How much, considering the very small amount of this material that's in the body of this individual and the very large amounts that go through an individual, what does the math look like? Now, this is what the math looks like. An adult takes about 900,000 calories of food a year off of this truck. There's about a 15-kilogram weight increase in an average adult human over the 25 to 55 years of age. There's about 5,000 kilocalories per kilo of body weight gain. Most of this is adipose tissue, but that's about the average for a kilo of body weight, which is 75,000 kilocalories stored of the 900,000 a year over the 30 years here. So it's a total of 900,000 kilocalories in times 30 years is 27 million calories, kilocalories, 75,000 over the 27 million, which is the excess that ends up getting stored as opposed to burned and used in heat and work, is a 0.3% excess of intake in terms of total calories over energy expenditure. Now, if you think about it, this is an extraordinarily close control, if you want to think about it that way, and I'm going to show you some of the physiology of how this is done. This is very tightly regulated. So this fits with the model that I suggested from evolution, that there are systems in place to keep energy balance pretty close to whatever is ingested. And 0.3% regulation, as you will know as well as I, is very tight for physiological systems if you compare it to things like osmolarity, serum sodium, or blood glucose. This gives evidence that this system is tightly regulated over time. Now, I want to take a little sidetrack here just for a second, because it will have occurred to one or another review that maybe this balance isn't quite the same if the individual's diet is radically different. In other words, if a person were taking more of these calories as carbohydrate or more as fat, maybe this tight balance that I'm arguing here wouldn't occur. And I just want to show you one figure from a study that we did many years ago, taking human beings who were in a clinical research center, eating only a liquid formula diet. That's only food they were fed. They couldn't go outside. They stayed in the clinical research center. And the diet was varied over time. This is days of the study, from one that had, all of them had 15% protein, one had 10% fat, 15% protein, and the rest 75% carbohydrate. Then the same individual was switched to a diet much higher in fat, much lower in carbohydrate. So here you have a seven-fold difference in the percentage of fat in the diet and an equivalent difference in carbohydrate. This is the body weight of the individual being fed those two different diets. There's no adjustment in calories. It's fixed in this case at 2145 kilocalories per day. It's no change in body weight. Now, I'm showing you one figure from this paper, but there were something like, 25 subjects that were studied this way, and the results were the same. So what I'm showing you is, despite all the storm and arguments in the literature about whether diet composition makes a big difference in energy balance, here you can see in a very tightly controlled situation, little or no effect of diet composition. What really matters is the number of calories this individual is eating, not the composition of the formula that the individual is being fed. So I want to try to get the issue of diet composition off the table here, or at least in terms of energy expenditure. In terms of energy intake, it could be quite different. Some people find one or another kind of diet much more attractive and tasty, and that's not what I'm arguing here. I'm just trying to get you to see, or at least to believe, that radical changes in diet composition don't necessarily have any effect on energy expenditure, because this individual is eating exactly the same number of calories of a radically different diet and not having any change in weight, which means the energy expenditure must be unaffected under these circumstances. All right. So here's where the rubber hits the road, so to speak, in terms of the clinical relevance of some of these considerations. Here's a figure from a paper 30 years old, but I could have shown you many, many different versions of this. These are individuals who are on very low calorie diets, very low calorie diet and behavioral modification or behavior therapy. This is a low calorie as opposed to very low calorie. You know this, actually, that individuals who are given a diet that is lower in calories than what they're expending will lose weight. So these individuals all successfully lost weight, or many of them did. But when the diet is either discontinued or the individual stops paying attention to the diet, the weight gain that occurs. So these individuals, despite being successful in weight loss and despite trying to stick to these diets, which are very difficult to do, ultimately 80 or 85% of people who successfully lose 10 or 15% of their body weight will return to their starting weight as a result of metabolic changes that I'm going to talk to you about. And if this problem could be solved, much of the so-called obesity epidemic would be addressable from either a pharmacological or a behavioral point of view. So in the US, there was this famous study called The Biggest Loser, in which individuals on television were actually paid to reduce their food intake and lose weight. And there was a whole sort of a weekly show that got a lot of attention. And what I want to show you here is that when these people were followed up, the same consequence resulted. So here you see really substantial amounts of weight loss. Up to the mean here was 60 kilos of weight loss. These are obese individuals. 6 years later, despite being very highly motivated to keep this weight off, they went back up to, in this case, minus 20, so that you can see they're on the way to regaining much of the weight that they lost. This is their sort of starting point. And this is the same for their fat mass, which was measured by DEXA. So what I'm showing you here is that even people highly motivated, not only to lose weight, which they did quite successfully, but to keep it off, this is something that's very, very difficult to do. So we've done studies over the years trying to address what is it about this reduced body weight state that seems to lead people inexorably to go back to where they started. So the studies I'm going to talk to you about were done in patients, individuals in a clinical research center, fed only liquid diet. Remember, I showed you a example of one of the studies that was related to that kind of strategy. And we would bring them into the clinical research center as lean or obese individuals, feed them the liquid diet until their weight was absolutely stable, and then give them 800 calories a day until they lost 10% of their body weight, refeed the liquid diet and amounts to keep their weight absolutely stable for another 6 or 8 weeks. And what we did at these 2 time points, before they lost weight and after they had sustained a reduced body weight, we measured all of these metabolic and behavioral phenotypes in these individuals, including functional magnetic resonance imaging of the brain, muscle and autonomic physiology, neuroendocrine studies. I'll show you a little bit of these data. But you can see what we were trying to do is to see what an individual looks like compared to him or herself after maintaining a 10% reduction in body weight. Simple, straightforward experiment. Not easy to do, I can assure you, because people being locked up like this, it's not a trivial sort of undertaking. Nonetheless, this is what we've done. And here is one of the first results of this study, which I think many people maybe don't sort of fully appreciate. So we measured energy expenditure in these people by knowing how many calories it took to keep their weight stable. We also measured it with room calorimeters and doubly labeled water. If any of you are interested in the methodology, I can describe it in more detail, but we measured energy expenditure in many ways, or at least several ways, and measured their body weight and their body composition as well. And so what we were trying to do is to figure out, what does the regression line look like of lean and obese people relative to their lean body mass? The lean body mass is the main determinant of resting energy expenditure and total energy expenditure, because the remaining part, the physical activity, is done by muscle. So total energy expenditure of lean and obese individuals, lean people being individuals who've never been obese, surprisingly to some people, are on the same regression line. So obese people, when they are obese, are eating, having to take in as many calories controlling for body size as a lean individual. They're not eating tiny amounts of food and able to maintain their body weight. They are eating an amount which is predictable from their metabolic mass and is the same proportionately as a lean individual. That's number one. And I think some people find this surprising, but nonetheless, this is the case. You might ask now, which is what I showed you in this weight paradigm, what happens when they lose weight, when we experimentally restrict them to 800 calories a day, get them down by 10%, and then measure their energy expenditure again, which is what we did. You might predict that they would just slide down this regression line so that you'd be able to predict what their energy requirement would be at reduced weight by knowing what it was up here, and the same for a lean individual. And that's not what happens. This is what happens. So when an obese individual loses weight, their energy requirement, their energy expenditure drops below this regression line that describes their energy expenditure when they're at usual weight. And when you do this to lean individuals, exactly the same thing happens. So at both starting body weights or levels of adiposity, you see the same physiological resistance to the maintenance of a reduced body weight. Now, remember in that first slide, I showed you you would predict from evolutionary considerations that the defense against weight loss would be strong because of the survival criticality of that particular piece of physiology. And here you see it actually represented in an actual experiment in humans who start at both lean and obese body weight. So where you start is not a determinant of the strength of the resistance. And we'll come back to this in a moment. When we look at the data from these, you'll hear me mention the residuals. And the residuals are the difference between what you would have predicted from the energy requirements of these individuals at usual or starting body hormone
Recording
would
in
cause
progress.
reductions in body weight, and it clearly doesn't have much of an effect in that regard in the average individual. And it was thought at the time that maybe obese people were resistant to the actions of leptin, but that lean people would respond. And that was why they were included here. And they don't respond any differently than obese people do. This resonates back with something I mentioned to you before about the fact that obese and lean individuals are regulating their body weights around the same homeostatic points, but the body composition that they're regulating it around is quite different. So this was evidence that leptin, at least in high doses, and here the doses are enough to raise the blood levels almost tenfold, really didn't have the kind of striking effect that was being hoped for. So high doses of this molecule or excesses of it don't seem to do much. But more interesting, as you'll see, is what happens when you have a relative insufficiency of it. So here's an experiment that we did now almost over 20 years ago in rats, in which we took rats that were either fed, starved, food restricted, or starved and given leptin injections at low doses, just enough to sort of raise the blood level of leptin back to where it would have been if the animal had not had the reduction in body fat that the starvation imposed. And so here, again, based on the little schematic I showed you earlier, as expected, levels of NPY and AGRP, the so-called orexigenic peptides, go up in the hypothalamus of these animals, and POMC goes down. And if you give leptin in low doses, you actually restore the levels of these peptides, both the orexigenic and the anorexigenic, to the levels that they were at when the animal was fed, even though the animal is still being fasted. So this, again, was evidence that what's happening, at least in part in the hypothalamus of a rodent at least, is that the weight loss by lowering leptin is setting off or triggering responses in the hypothalamus that would be expected, and they do drive the animal to seek additional food. These animals also show a reduction in energy expenditure, which also, given some of the other physiology that I showed you or has been shown about leptin, is fully anticipated. Rexahema around this time showed very similar responses to other hypothalamic axes in mice that were underfed. So this was a consistent finding with remediation or reversal or partial reversal by administration of low doses of leptin, suggesting that leptin deficiency is at least part of the mechanism for these changes in hypothalamic gene expression. So here's the protocol that I mentioned to you before. And as you might imagine, now what we did is we took these same individuals who had been maintained at reduced body weight, and we did those many physiological studies that I alluded to in terms of energy intake and expenditure. And we now gave these humans leptin at doses very low, not like the ones that were done to try to provoke weight loss, but only at amounts sufficient to restore the circulating concentration of the hormone back to where it was in these individuals before they had undergone this 10% reduction in body weight. And obviously what we were interested in seeing is whether or not this would reverse the metabolic and behavioral phenotypes that were invoked by the loss of body weight and necessarily body fat. And here we did the same measures, some of which I alluded to earlier. And here's a summary of the results of those studies. So I mentioned the energy expenditure, the energy intake phenotype. We've done functional magnetic resonance imaging of these people while they are viewing food and non-food items so that we can gauge their response to food when they are both at usual weight, weight reduced, and weight reduced plus a low dose of leptin. We studied the autonomic nervous system, neuroendocrine axis, as I mentioned, and these muscle phenotypes. And what I want to bring to your attention, and I'm not going to show you all the data to support this, is that the administration of this very low dose of leptin, just sufficient to restore the level here to what it was here at the start, actually makes all of these phenotypes go away, or substantially or totally go away. So it corrects the energy intake problem, the energy expenditure problem, including the muscle physiology, the neuroimaging that shows changes in both frontal cortex and other parts of the brain related to motivation and reward. It rectifies those. It restored the autonomic nervous system, with the exception of the parasympathetic nervous tone, and restored muscle physiology, with the exception of one of the sarcoplasmic reticulum calcium gates. But that efficiency change that I mentioned, and the preferential use of fatty acid or reduced glucose utilization, rectified by low dose leptin administration. So one way to think about this is think of it, again, in a very simplified way, as an electrical circuit in which leptin, at least in this case, and remember, I'm focusing on leptin, but there are clearly other players in this system, acts as the sort of current, the electron source, if you will, that is driven through a series of resistors, in this case in the hypothalamus and other parts of the brain. And here I'm just showing you a couple of the canonical receptors in this system, like leptin receptor, melanocortin-4, cannabinoid receptor, but GLP-1. There are many others that could be in this. But what I'm trying to show you is that this circuit can be pictured as a circuit in which, if you do things to the status of these receptors, either genetically or by virtue of acquired differences in their function as a result of weight perturbation, you can get changes both in the neuronal activity and glial activity that can affect the efficiency with which this circuit engages. And what I'm saying to you is that when you drop the leptin concentration in an individual as a result of weight loss, this circuit now is not seeing, however it's set, either for an obese or a lean individual, the amount of current that it is ordinarily expecting or anticipating, if you will, and that by virtue of that lowering, and also other metabolic changes that occur under these circumstances with lipids and cytokines, you basically have a circuit that cannot sort of fully be completed with consequence with regard to the regulation of both energy intake and energy expenditure. Another way of looking at this, as we want to consider that sort of circuit as a model, this line here represents what might be called, or what we consider, the threshold for the action of a hormone like leptin. And again, there are others clearly involved in the system, but obese individuals with this kind of model have a threshold which is set higher than that of lean individuals. And the setting of the threshold, as I mentioned, is a result of genetic factors, developmental circumstance. We know that perinatal and prenatal physiology can influence brain development in a way that probably affects the level or the location of this threshold, and these other cell types that I mentioned earlier can influence where the threshold is located. But the point I want to make is this, that an obese person's threshold is higher in this model than a lean individual who has less body fat. And all that's required for the system to feel that it is in sort of physiological homeostasis is to have enough of the signal coming from this tissue so that it is above this threshold. And then the organism is eumetabolic or homeostatic. And the problem with weight reduction in an obese individual or a lean individual, I just didn't schematize them here, the obese individual's fat mass is reduced to the point where the signal, which is proportional to the amount of fat, is no longer sufficient to get over this threshold. And again, we'll come to this right at the end. The threshold cannot be changed ordinarily except by mechanical interruption, neurosurgery or something, so that if you lower a person's body fat, you're actually putting them into a situation in which they sense this as a deficiency state, in the same way that an obese mouse is hyperphagic because it's not getting any leptin signal. An obese individual whose weight is reduced below this threshold point, the signaling is below the threshold point, this individual will behave as if the survival was threatened because inadequate fat stores are available for that particular threshold. And as I showed you, if you give excess levels of this hormone to individuals who are already obese or lean, there's minimal or no effect. The physiology is in the circumstance in which the level of the hormone is reduced below what this threshold is anticipating. And as I showed you, at least for this model, the body doesn't adjust or compensate. It continues to show this physiological adjustment. So in the last couple of slides, I just want to sort of revisit this with you. So here is this idea of a threshold where it's looking for a leptin concentration, which is generated by a fat mass. So the fat mass increases, the leptin signal goes up. This is well known. I mean, this relationship is classic. So the question is, what can be done experimentally or physiologically to raise the threshold? You might not want to do that, but I'm just saying experimentally. So if you make a lesion of the ventromedial hypothalamus of a rat or a mouse for that matter, the animal will behave as if you had raised its threshold. It will begin to defend a higher level of fat mass. Maybe some neuronal changes with aging, possibly at puberty and pregnancy, there are changes in where the threshold is at least transiently located that cause body weight, a higher fat mass to be sought, if you will. A critical question is whether chronic weight increase can raise this threshold. In other words, if somebody who would ordinarily be at a body weight of 70 kilos maintains a body weight of 100 kilos due to environmental factors, can that individual's threshold be pushed up over time so that they will again begin to defend 100 kilos rather than 70? This question has not been adequately answered in humans. It's probable that under extreme circumstances, this can happen. But I think it's not that common. I don't that people will have their threshold raised environmentally. Can the threshold ever be lowered? In other words, can you cause a person to want to maintain a lower body fat? And cachectic illness does this. Lateral hypothalamic lesions in rats will do this. They will permanently defend a lower body fat. And anorexia nervosa is clearly a physiological state in which, at least behaviorally, a lower threshold is being maintained. But as I showed you, and this is the critical point for this talk, prolonged weight reduction does not lower the threshold. You can maintain a reduced body weight for 6-8 years, and there is no evidence of relief of the behavioral or physiological adjustment to reduced body weight. Could prolonged weight elevation raise the threshold? I mentioned that. It's not entirely clear. I think in some cases it could happen. It's probably not that common. So you're undoubtedly thinking about, or have been thinking about, some of these newer GLP-1 receptor agonists, and I just wanted to end by pointing out something to you. So here is a figure from a very recent paper on this, on tirzepatide, which is a GLP-1 and GIP combined agonist. Really very striking amounts, this is a 20% weight reduction. I mean, this is a very impressive pharmacologic effect here over a period of 36 weeks. And then what they did in this study is they stopped or put these people on a placebo, and exactly what you would, or at least I would, and maybe you should predict will happen, is that the weight loss effect doesn't persist. These guys begin, this is a follow-up period of up to 88 weeks, so it's another 50 weeks or so. These people are probably on the way to regaining the majority of their lost weight. They're now down by only 9%, when here they were down by 20%. If you keep giving the drug, they keep losing weight beyond this point. That's not a surprise. But even then, they ultimately level out, suggesting that these people have now reached the minimum that, at least at this dose of these drugs, their body fat will tolerate, or the brain will tolerate, with regard to their body fat. Otherwise, if this were just an open loop, you would expect these people to keep going down until they basically starve to death, which isn't what happened. So there are two messages here. One is that there is a equilibrium that's reached even with these very powerful agents, and we're actually getting ready to undertake some studies to look at what happens to the energy expenditure of these guys down here. I don't think it's hard to predict that they're going to be in a more energy efficient state. But also, very strikingly, if you stop these drugs, and I'm sure this is going to be true of all of them, you're going to get a rebound, because the physiology that I described to you is in place no matter how you manage to get the weight down, either by diet or with one of these agents. So I want to end by pointing something out to you that it's probably now occurred to you. So if you take people who are in their usual or eumetabolic state, this is obese or lean, and you drive their body weight down by 10, 20, and %, they become hypometabolic and hungry. This is not a normal physiologic state. I call it an induction of a non-physiological state. And I think one of the critical requirements, the critical necessities in the area of human obesity and its treatment, and maybe ultimately its prevention, is to recognize this fact that people are going to try to resist what otherwise may be cosmetically or clinically a very favorable new body weight by virtue of being hypometabolic and hungry. And the majority of them are going to go back to where they started. But what I want to point out to you is that it's not that hard to restore their normal physiology. In other words, the ease with which this can happen is shown here by the thinner, smaller arrow. And I showed you one way, at least one model for how this can be done, which is restoring the circulating concentration of at least one of the hormones that is the mediator, or is among the mediators, of the body's mechanisms for sensing how much body fat it has. So with very little leptin, we were able to essentially reverse this hypometabolic and hungry state. And this, I think, is something that we ought to be paying a lot more attention to among individuals who work in this area. Obviously, we need to know more about the neurobiology and so forth. But this, I think, is a very critical and important point, is that the physiology of getting somebody's weight down is very different than the physiology of restoring their metabolic or eumetabolic state pharmacologically. And I'll show you or end with this one slide that makes this point. This is an experiment we did a number of years ago with a melanocortin-4 receptor agonist that belonged to AstraZeneca. And the details here are not critical. I just want to point out something to you. So we gave the dose by osmotic mini pumps, and here you can see the animals that were treated with this losing weight and maintaining a reduced body weight for a period of time. They were still on this high dose of the melanocortin-4 agonist, here shown in yellow. And here we had a group of animals who we reduced in body weight by restricting their caloric intake, and then either put them on a low dose of this melanocortin-4 agonist, literally like a fifth of the dose that was used to cause the weight reduction. That's in the black line here. And you can see the animals, the controls went back to where they started. They were treated with vehicle. But if we gave a 1/5 of the dose of the melanocortin-4 receptor agonist, we could maintain those animals at the body weight that in order that would when compared to animals who got the drug as a therapeutic weight reducing agent, we could get that maintenance at 1/5 the dose. So I take this as evidence again with an entirely different molecule and receptor system that low dose replacement can be as very, very effective when compared to much higher doses required to get physiological weight reduction. So I think we ought to be thinking about this a great deal in terms of the pharmacology of the weight reduced state, not just what agents are effective, but the almost certain situation or circumstance that we'll be able to get away with much lower doses of these agonists or these agents. And I want to mention before I stop, this is the last slide, that we're actually doing a study now, an NIH study, looking at individuals before and after a 7% weight loss with regard to, I'm not going to go into the details of this, a wide variety of physiological studies of them in the process, for most of them, of regaining their weight. This is done by diet, and then they're encouraged to try to stay on the diet. We know that many will have trouble with this, and we're studying them as they regain their weight using many of the techniques that I alluded to in the studies that we did with leptin to try to prospectively understand where the most salient touch points for trying to prevent weight regain can be found. Because again, as I've tried to emphasize, the problem is not so much in getting people to lose weight, it's in getting them to maintain the lost weight. And even modest success in that regard will have a big impact on many of the comorbidities of obesity, as again, you're undoubtedly aware. So this is the so-called POWER study. If any of you are interested in it, you can look it up on the web, or you can write to me and I can tell you more about it. We're recruiting subjects in New York and Philadelphia for this now. So I just want to make sure to acknowledge the people who did a lot of the work that I described. Mike Rosenbaum, who's been doing the human studies with me for now over 30 years. Rochelle Goldsmith is an exercise physiologist. Jules Hirsch, very well-known physiologist of human body weight regulation. Wahida Karmali did the diet manipulations for us in those inpatient studies. These are individuals known to some of you who have participated in various ways in other studies going on in the laboratory, but not the human studies per se. This is Louis Aroney, who has collaborated with us along with Martha Heener in studies of skeletal muscle biology in the weight regain state. Here's somebody many of you will know. These are other people who have participated in these studies. Wendy Chung, who's done a lot of genetics on these, and Charles, who has studied the physiology of the mice that have undergone those weight perturbations that I showed you. And here are some others. Robin Goland is a clinician who has helped with the recruitment of many of these patients. Stem cell biologist known to some of you who are in the group. And a psychiatrist who's very interested, again, in body weight regulation and weight regain. Dipna Gallagher is a body composition expert who was one of the PIs of that POWERS weight regain study. So thank you for your time and attention. If you want to get in touch with me, my email address is here. And now I'd be happy to hear from any or all of you with regard to any questions and advice you've got for me. Thanks.
Okay, and Well, thank you, Rudy, for this wonderful talk and a lot of informative information and pretty yeah. So we will have actually a Q&A section with our panelists, and then all the audience, if you have questions, please write down or you can raise your hands. And we will have our first panelist, which is Dr. Li-Heng Wang, and also she is Dr. Rudy Leibel's own student and very successful, well-trained, and she's currently in Beijing University. And Li-Heng, yeah, please unmute and ask. Yeah.
Hi, Rudy. It's great to see you in this way. So actually, I have 3 questions. Actually, I may have more, but I save time for the other audience. So my first question is, what determines the threshold? As you mentioned, like some genetics factor and also the circulation hormones and also many others. But what I mean, because I have heard your talk for many years, I just wonder whether there are a clear answer for this question. What determines the threshold, particularly, I mean, the body weight set point that you mentioned?
So this is the critical question. When I showed you my sort of electric circuit and then that threshold figure, the short answer is I think that obviously genetic factors play a major role in this. If you look at again, these famous twin studies that have tried to apportion the risks of obesity or excess body fat to genetic and environmental factors, it is thought based on those studies that genes probably convey somewhere around 40 or 50% of the risk. And I think that's the reason why I showed you this sort of electric circuit here, which is obviously greatly simplified, but these genes there are many of them. At least there's over 100 that have now been identified. They're not all receptors. They don't all play in the hypothalamus. But I think a major part of this has to do with the wiring of the central nervous system, which is under genetic control. But here I'm mentioning other factors, such as early and prenatal and early postnatal effects of diet and other environmental influences that are changing the structure of the central nervous system in a way that ultimately locks in so that you get this sort of what you referred to as a set point, or we refer to as a threshold, because I think the strength of its resistance to weight gain is not as strong as its resistance to reduction in body weight. But whatever you call it, I think that this line is established by impact that is conveyed by many factors, with genes probably being the single major influence, but clearly not all of it.
So I think my following question is kind of related, because you mentioned the initial body weight, and then the weight gain, like when you become obese or you have when you lose your weight. But so what really determines or defines the initial body weight? Because take my personal example, for instance, when I was at college and maybe 50 kilograms, but then when I go to when I'm reach my early 30s, I become maybe 55, and now I become 60 kilograms, and I couldn't lose my weight. So what really, I mean, if I define my initial weight as 50 kilograms, then right now I think my set point is changed. But according to your talk, the set point cannot be changed. So I want to hear your.
Right. So the question is, in somebody like yourself who has had an increase in body weight over a decade or so, would your response to weight reduction be the same as the individuals I showed you who are chronically lifetime at that weight? and we don't know the answer to that. That's the kind of experiment that needs to be done. I would predict that you would not show as much or maybe any of the metabolic compensation that one of these individuals who had habitually been at that body weight would show. But they're clearly, and that's what I tried to imply in my sort of earlier comment, there probably are circumstances in which this threshold or set point can be elevated. But I think in the majority of instances, people who have gained weight as adults are reflecting their interaction with the environment, which has overwhelmed whatever resistance there is to weight gain. And as I've tried to emphasize, from evolutionary point of view and physiologically, the resistance to gaining weight by this system is not nearly as strong as the resistance to weight loss. So you've asked a very important question, and it's very hard to answer, because what we need is people who have gained weight over various periods of time to test whether or not any of them are now starting to stick in a set point sense at that higher body weight. We just don't know.
Okay, but in a particular study, the initial weight is defines at the start of the study, the start point of the study, the people's body weight. That's how those studies that defines the initial.
That's correct. So what we do is we recruit people who have habitually been at that body weight for 4 or 5 years or more. In other words, we want to protect these studies against the question that you just asked, because that's a different question. So we're trying to get people who are at their habitual long-term body weight or body fat, and that's where we get the obese and the lean individuals from. We don't want to study, or have tried not to study, people who've had shorter term changes in body weight, up or down.
Okay, so my third question is actually related to the evolution of the body weight defense mechanism, because you just also mentioned the defense against the loss of fat is much more than the defense of body weight gain. So I can understand that in the past because of the surviving issue. But in right now, because people we have enough food for consumption, I mean, most of us do not need to worry about hungry anymore. So I mean, so from the evolution aspect, as the time goes, you think whether this will change along the time? And also, I want you to know the theory or the mechanism behind that. Why was the difference?
So I think the difference that I described is due to the fact that environmentally, if you think 100,000 years ago, our forebears, our ancestors out of Africa, so to speak, what they had to contend with was almost always circumstances in which they couldn't get as much as they would have perhaps have liked to eat. I think they were existing in a circumstance of restricted access to calories. The opportunity to get obese was probably very, very limited. So our genetic pool, if you will, which, is only, if you want to think about it that way, 100 or 200,000 years old, it hasn't had time to change, if it were going to change in a response to evolution, to give us better defense against excess weight gain. And one of the reasons for that is, first of all, it takes time. And I don't think it would have happened because the consequences of excess weight gain in our current environment are not enough to interfere with, in a major way, with reproductive efficiency. And we have medical approaches that will mitigate the consequences of the higher body fat, so that those individuals are protected now. They're not being subjected to the pressures of evolution, which would have to occur in order to select for individuals who were resistant to weight gain. There are such genes in our gene pool, and I didn't discuss them here, But there are a few genes that look like they don't look like they're G protein coupled receptors, the ligand for which is unclear, that actually do defend against weight gain, but the number of those and their potency is much less than the genes that defend against weight loss.
Okay. So yeah.
I don't think evolutionarily it'll ever happen at this point. I don't think.
Yeah. It takes longer. The evolution.
It not only takes longer, but those people are protected. They're not dropping off out of the reproduction pool, which is how evolution happens. There has to be a selection against them, and it's not happening. I mean, the environment just isn't doing that.
Yeah. So I have one more question regarding on the last piece of data that you show is about the MC4R agonist treatment. Mm-hmm. Your opinion is to use once you have first like a 20% weight reduction, and then you give a low dose versus at the initial you give a high dose of the agonist. You think the low dose works better, but just from that, actually there are two small questions came out. One is what drive the weight reduction? Does that matter if you are using the same reagent to reach the weight reduction, but the people or the mice may become like the drug resistant? And if you lower the dose, you cannot see the kind of effect that you see in the animal. But then from another perspective, does that mean that the combination of different weight reduction reagent, like two different, like the drug that, to give in at a different time point, with the combination, you can use a low dose to achieve the same effects with kind of a single drug with a high dose in the weight perturbation. Yeah.
Yeah, I understand your question, and I think it very much will depend on the pharmacology of the agents being used. But I agree with the premise that when we now, at least what I'm trying to argue, and you've picked up on it, is that the pharmacology of management of body weight in humans should not be a sort of a monolithic idea that we give a drug, we try to get them to lose weight, and then what do? If we stop the drug, they're going to regain the weight. It may very well be that we will use different agents to achieve both the weight loss and the prevention of weight regain for just the reasons that you mentioned. Some may turn out to be much more effective at provoking weight loss. The GLP-1 agonists are certainly in that class, but it may turn out that other drugs, maybe some that have failed in the clinic for weight loss, will be very effective at maintaining reduced body weight. Leptin itself may be an example of that, where it doesn't do anything really useful in terms of clinical weight reduction, but based on what I showed you, may be quite effective at maintaining reduced body weight that you reduced by means that were not dependent on that particular molecule. So I agree with the premise of your question. And if I provoked your thinking that way, then I feel very pleased that my talk was effective, because that's exactly, I think, the way we ought to be thinking about this. We ought to think about the process, the mechanisms, the responses to weight loss, and think about therapeutic interventions that may not resemble, either in terms of the pharmacology or the biology, the process of weight loss itself. I absolutely agree.
Okay, thank you, Rudy. So pass this to other panelists.
Thank you, Li-Han, for all these tough questions and great questions. We have our next panelist is Dr. Li Qiang, so from Peking University.
Okay, so Rudy, great, and I'll keep it brief. Start with one general question. So like you showed, our body is designed, or in our lifetime, designed to consume 3 times of protein, 3 times of carbohydrate. So if we eat more, consume this faster, that means that will shorten our lifespan. So our body cannot consume that much. So in the same way, calorie restriction will have this anti-aging effect?
So, the short answer is I don't know. And my longer sort of response or hypothetical response is, I think from the point of view of the energy homeostasis system that I'm showing you here, pretty much the way to think about this is not to overthink it. I think that in this system, calories are pretty much calories, no matter how you time it or take it. Now, I know there are studies showing that if you restrict feeding to a certain number of hours a day, you can see differences in weight loss and weight maintenance and so forth. But I think over time, the metabolic compensations that I'm talking about here, where the body fat and other parts of the body begin to talk to the rest of the physiology, will make it difficult for those interventions, if you want to call it that, to have long-term effects. It's clear, at least I think in some of the animal studies, that restricting calories chronically will do something to the longevity of these animals. But again, what is shortening their longevity in some instances is tumors that are not necessarily analogous to what happens in humans. And again, humans who are subjected to chronic restriction in calories, which some people are able to do, are generally very uncomfortable. So even if it turns out that in humans, by caloric restriction over extended periods of time, you can do something to enhance longevity or prevent some of the metabolic consequences that would otherwise occur, people may be so dysphoric as a result of being put into that situation that the extra time won't be worth it. I mean, I know that's a sort of a smarty sort of answer to this, but I think current thinking about this, or at least my thinking about it, is to pay close attention to the thermodynamics and to treat a calorie pretty much as a calorie, regardless of the circumstance in which it's taken.
Okay, so thank you, Rudy. My second question is related to the GLP-1 drug. And your theory many years predicted with the body weight reduction, there is a loss of energy expenditure due to reduced muscle mass. That's exactly the problem of the GLP-1 drugs. So now the people or the company trying to combine some therapy to regain or maintain muscle mass. So in this way, the muscle mass may be not equivalent to muscle quality or activity. And also, my other thinking is, so this against our threshold, body threshold setting, so maybe it's not a physiological or healthy. I want to hear your insightful comments on this. Thank you.
So let me make one point about the muscle mass that you mentioned. In this schematic, I'm sure you guys can all still see this, right? So the point here is this, that when a person loses weight, they do lose lean and fat mass, both a lean and an obese person. On average, the weight loss is about 70 or 80% fat, and the rest is lean mass or muscle mass primarily. But this figure here is trying to make the point, and does make the point, that it's not the reduction in muscle mass per se that is slowing the metabolism. That definitely happens, but more striking is that the muscle that remains is more metabolically efficient, or at least more efficient in terms of muscle contraction. So you're right, loss of lean mass reduces energy expenditure or total energy expenditure, but the compensation that I'm talking about is in spite of, or in addition to, the loss of lean mass. The performance of muscle is altered so that it has this more efficient phenotype. Now, what will happen in some of these strategies that you alluded to try to restore or maintain muscle mass under these circumstances, I don't think obviously anybody knows. It may be helpful, and whether or not the additional muscle mass that's retained or the muscle mass that is not lost will have this metabolic phenotype or not, I don't know, but it's possible that even though you retain or maintain some lean muscle mass or muscle mass, it will still show this metabolic compensation. So you may not get as much benefit from maintaining muscle mass as you might think you would, because that muscle will show the same metabolic compensation that the muscle mass, the endogenous muscle mass, had as a result of weight loss. I know what you're talking about, but it's not, the point I'm making is that even the remaining muscle is showing this increased metabolic efficiency, and what the consequence of giving a myokine or some other agent to support muscle mass will be under these circumstances, I think it's very hard to predict. It may be beneficial, but it may not have as big an effect as people hope it will because of what is going on in the muscle mass that remains.
Thank you, Rudy.
Yeah.
Actually, our next panelist is Pingwen Xu from UIC.
Hello, this is Ping from University of Illinois at Chicago. Fantastic talk. I really learned a lot. And also, I very interested about this theory, like every person have a set point for body fat, and any deviation from set point will produce compensatory
has
adjustment.
a set point for minimum body fat. I think that's an important point. So there's each person has a minimum below which this process of compensation will be invoked.
Okay,
anyway,
got it.
Go ahead. Yes, I get your point.
Got it. So based on the data, leptin is obviously involved in the process of this rebound effect, and those low dose of leptin supplement can prevent body weight regain after body weight loss. So is there any existing clinical trials trying to supplement the low dose of leptin or other related potential hormones after GLP-1 agonists withdraw in body weight loss trials? So that could be an interesting topic.
Yes, I agree with you. And again, I believe, some of these things don't get published, that an effort was made to try leptin in weight-reduced individuals, but my understanding is that the doses that were used were probably too high. So it might sound strange, but based on what I said about efforts to treat obesity with leptin, maybe you would believe this, is that if you give too much of the molecule, the central nervous system will reduce its response. So you need, ironically, under these circumstances, to give doses that are very small. Like when we did the studies that I mentioned in humans, they couldn't tell that they were getting either leptin or placebo. Normally, at higher doses, leptin stings when it's injected. It has a cytokine-like effect locally. So the short answer to your question is, I think if it's been tried, it was probably tried at doses that are too high. But if what I've done has gotten you to thinking of other drugs, leptin may be one of them, but given at doses that may be very different from what would be required to get weight loss, I think that's exactly the kind of thing we ought to be thinking about. And the way that we study these drugs in animals almost always is somebody will give the drug to a lean animal, they'll give it to a high-fat diet-fed animal, but never give it to a weight-reduced animal to see whether or not it has the effect of maintaining a reduced body weight, which is physiologically very different, as I've tried to point out, than driving body weight down. And you never see these studies. I mean, I've certainly written a lot of papers about mice. I've written a couple, actually, as you might imagine, on weight-reduced mice. But in general, you don't see this in the literature. And I think we need to think more along these lines. When you have an interesting molecule, test it not only as a weight reducing agent, but as a weight maintenance agent, and you almost certainly at doses that are generally going to be much lower than what you would have to give to induce weight loss. And that may make agents that would otherwise not be attractive for weight reduction very attractive for maintenance of reduced body weight. That's exactly right. I mean, I agree with the premise of your question.
Thank you so much. That sounds like a R21 project already.
There you go. Yeah.
My second question is related to sex differences in this set point. It's obviously female have a lower set points compared to males, and also females after menopause, their set point also changed. So they after menopause, the females have increased of body fat and a decrease of lean mass. So apparently, there's something happens during the menopause. So the set point increased to a certain level. And also there are some clinical trials showing that the estrogen replacement therapy can effectively prevent this change of set point. So do you think this estrogen sex hormones could be one way to change the whole set point?
I didn't show this slide, but maybe I should have. That we know that at any given level of body fat, if you measure body fat by DEXA or underwater weighing or MRI, women premenopause in any event have higher levels of leptin per unit of body fat than men. And this is due to the fact that subcutaneous fat, which is predominant, or women have a larger fraction of their total fat mass subcutaneously, produces more leptin per cell than visceral adipose tissue for reasons we've not been able to figure out, but this is nonetheless true. So women, if I were to show you a plot of females versus males, you would see that the women have higher leptins per unit of body fat than men do. And actually, when they enter menopause, there's some decline as a result of the fact that they actually begin to acquire more visceral adipose tissue relative to their total fat mass that actually produces, as I mentioned, less leptin per unit of fat mass. So you're right to ask the question, why is it that women are set at a higher level of body fat than males? And again, I would answer this in an evolutionary sense, that they need the extra body fat in order to be able to successfully carry a fetus. That costs about 100,000 calories. So I think evolutionarily speaking, women are designed to carry extra body fat as, at least in part, an evolutionary response to the need to be able to feed a newborn for a period of time, and the energy cost of the pregnancy itself, which again, not now, but 100,000 years ago, the environment was a much more hostile potentially than it is now. So they're actually built to be able to sustain higher levels of body fat, and they defend that higher level of body fat. I didn't mention this, but there were women in the study as well as men, and they behave in terms of defense mechanisms, these energy responses, in exactly the same way that men do, or men respond exactly the same way that women do, but they're defending different amounts of body fat. Now, what it is that actually permits the female to do that is, I think you're on the right path, but again, we don't know the answer to this. I would believe that at the time of puberty, for example, the changes in the gonadal hormones that occur in a female change the central nervous system wiring and sensitivity to these various molecules in such a way that they then begin to defend higher levels of body fat on a physiological basis in order to make them able to carry pregnancy successfully. But what it is, and you're right, I mean, again, some of this is known. I mean, it's known that estrogen actually changes the wiring in the central nervous system in parts of the hypothalamus and elsewhere in directions that are not inconsistent with what we're talking about. And I think in a very broad sense, that's the partial answer to your question, but the details, I think, again, are not known. And I mentioned in one of the slides, pregnancy is a very special circumstance in which a female needs to be enabled to take on additional stored calories as part of this process of being able to carry a pregnancy and feed a newborn. And again, we don't understand that biology very well either, but it appears to be much more transient than the process that we're talking about with regard to, for example, estrogen imposing a permanent change on the threshold or set point. Now, these are all very interesting questions that I can't answer. So that means they're good.
Yeah. Myself is a endocrinologist. I'm studying brain and estrogen effects.
Yeah, no, I think this is the question is absolutely on point, and we just don't understand this well enough to be able to answer the question. But I think in a very broad sense, these are all, at least in part of a system that's designed to enable a female to be able to sustain and carry larger amounts of body fat during periods of reproductive competence.
Thank you. Thank you so much.
Sure.
Thank you, Ping, and all our panelists. And our next, we will move to the chat question, and we see quite a few. And we still have 150 audience here, so it's great. And we will start from Mingming Wang. So, Dr. Leibel, I wonder if you have any suggestions on how people can set up phenotypic screen to look for agents that may restore the new normal after weight loss. I think we briefly touched, but
so if maybe I'm not restating the question properly, but how to get the physiology, what I describe in this one of these slides here. Let me see if I can find this again. So one of the questions I'm trying to get you all to think about, and maybe it's been successful, is the fact that an individual who has lost weight in terms of their own metabolic status, if they are eumetabolic at whatever body weight they were, and I've tried to point out to you that metabolically, if you adjust for body size and body composition, obese and lean individuals are superimposable. They're on the same regression line, but both achieve what is for them at least an uncomfortable and might be called a non-physiological state with regard to metabolism. They are energetically more efficient and have an increased drive to eat. And what I think, and again, is one of the critical questions in the field is, and I think that's what this question is about, is how to restore normal physiology without having the person regain their body weight. If they regain the weight, they go back to being perfectly in balance in terms of their hunger and their energy expenditure, but they're now back in some instances at the higher level of body fat that they found either cosmetically or clinically unhealthy. So this is a critical question, how to restore the normal physiology without having to restore the body fat. That's the critical question. And I've tried to show you at least one approach to this, which is basically trying to trick the brain that the body fat is not lost. That is, that you just basically put some of the hormone signal back into the individual. I'm certain that there are other ways to do this that engage the various elements in the central nervous system even more directly than this does. I mean, this is basically restoring a peripheral hormone. I'm certain that the circuitry, or at least I'm convinced that the circuitry in the central nervous system is also altered, as I tried to imply by that little simple circuit schematic in a way we just don't study this. I mean, again, I've said it once, I'll say it again. You know, all of us write papers about what happens in the central nervous system that can cause reduced energy intake, increased energy expenditure, but we don't study these animals or humans, for that matter, very much when they are in this state that I've described as non-physiologic, to try to understand what might be done to restore their physiology without having to have them regain their body weight. So to the extent that what you're asking is how to do this, I think it's a critical question. And you don't see very many experiments anywhere in the literature that are trying to address this question. So if you've got ideas, I mean, you know, I think you could look almost anywhere in the central nervous system and you would see evidence of changes that are the result of the maintenance of a reduced body weight. And there would be a variety of experimental strategies for trying to manipulate the central nervous system, either pharmacologically or molecularly, to see if it could be adjusted in such a way that this metabolic and behavioral compensation would be mitigated.
Okay, thank you. And so the next question from Chun Xue. And the question is, can you please comment on the pros and cons of drug candidates to induce lipolysis?
The idea of inducing lipolysis as a way of losing weight, I call your attention to this. So if you want to change the amount of stored energy in the body, which is, for purposes of discussion of obesity, is body fat, the only way to get this to happen is either to take a syringe or a needle and suck the fat out. I mean, people have done that. If you provoke lipolysis, what you're doing is taking the energy that's stored here, this internal energy, and you're asking that this part of the system spend it. It has to go somewhere. In other words, just inducing lipolysis itself cannot reduce body weight. You've got to burn those calories somewhere. So my short answer to you is trying to stimulate lipolysis is not the answer, because you won't, unless you do something special, you won't drive energy expenditure up substantially by doing that. You might argue that maybe by doing it, you would suppress energy intake due to the effect of fatty acids and other metabolites from adipose tissue. But the only way that you would get a therapeutic effect is by having some impact on these. Lipolysis alone is not a way to reduce body weight. If you can convince yourself, and I'm not really convinced that this is a particularly efficacious way to do it, that you would reduce energy intake and increase energy expenditure somehow to an extent that would account for the amount of body fat you want to lose, it would be therapeutically helpful. But for the reasons I just mentioned, I don't think it's a good approach.
Thank you. So the next question from Guo Zhang. A nice talk. In terms of the weight reduction trial, can you please comment on its effect on the plasma metabolism? Thank you.
We and others actually have not carefully studied the metabolome of these individuals, but this study that I mentioned to you will. So this study that's now going on, we will study the so-called metabolome of these individuals before they undergo weight reduction, after they have achieved this weight loss, and in the process of trying to maintain it for 12 months, we will generate the data that you're asking for. It's really, at this point, not sufficiently studied to be able to give you a competent answer. It's an interesting question, and this study is designed in part to answer that. But right now, I don't think we know enough to be able to answer it. I mean, I can obviously tell you some of the things that change in terms of levels of metabolites like glycerol and fatty acid and glucose and insulin. I mean, we know in a global sense what weight reduction does to those phenotypes, but I'm assuming that you're asking about other small molecules and metabolites that have really not been carefully studied under these circumstances. You need very careful control, not only of the body weight of the individual, but of their dietary intake and their the stability of their body weight. It's a difficult question to answer, but this study will hopefully get some of that information.
Okay, so the next question from Chunxue is same person, I guess. Is it physiological possible to lose fat mass selectively but maintain or gain lean mass?
Yeah, so Lee was asking about this. So the short answer to your question is if you put people on diets, you know, hypoenergetic state where their intake is less than their energy expenditure so that they necessarily lose weight, on average, as I said, about 20 or 25% of that weight loss is lean mass, and the remainder is fat mass. But within those sort of constraints, we don't really know any way to get preferential loss of fat over lean. There's some interest in some of the other peptides that circulate, like glucagon and other congeners of GLP-1 that may be able to influence the relative rate of loss of one or the other of these. But again, this is not clear at this point. By and large, I think standard conventional weight loss, either by drug or diet, will produce roughly the same fractional weight change, meaning 75% fat and the rest lean mass. Now, when you regain the weight, you get the lean mass back, but you also get the adipose tissue. So what Lee was talking about is a pharmacologic stunt that would preferentially restore lean mass without having to increase fat mass. And again, in theory, that is possible, but what the quality of that muscle mass invoked by a myostatin-like agent, for example, would be, I just don't think we know.
Okay, so and I think the next question is related to muscle. So from Dongdong Wang, thanks for the great talk. And could you comment on what kind of proteins or pathways could be used to increase energy expenditure in muscle after weight loss?
I can't find I thought I had the slide here. I was going to show you. So we did a study in which we took individuals who had lost weight and studied the effects of that energy expenditure phenotype, which I mentioned to you several times, this thing here, where you get the increased work efficiency. And as a result of the preferential appearance or the relative excess of slow twitch muscle fibers. So we did an experiment with Lou Aroney and Martha Heitner, that I showed you her picture before, in which we took a group of individuals and had them do resistance exercise after losing weight by putting them in a gym. And rather than having the sort of standard, which people do by walking or running or doing things to increase their energy expenditure that way, we had them do it by resistance exercise. And what we found actually is that if you have a person do resistance exercise, you can eliminate or reduce the amount of the increased efficiency. So sounds kind of, it's interesting. What you're actually doing is restoring the fast twitch muscle fibers, which are glycolytic as opposed to fatty acid burning, less efficient in terms of their coupling, mitochondrial coupling, if you will, or their chemomechanical coupling. So the short answer to your question is yes, and resistance exercise may be the way to do it. So when you have individuals who have successfully lost weight, one of the exercise prescriptions that should be carefully considered, rather than the sort of standard of keep walking or running or whatever you've been doing, is to have the individual undertake an exercise protocol that favors or has a high amount of resistance exercise in it. That will probably reverse some of these energy efficiency or these coupling efficiency changes that occur in muscle. And when you look at the muscle, you can see evidence of reversal of these phenotypes. And it's known, that resistance exercise has this effect on skeletal muscle anyway, that it will tend to favor fast twitch fiber development. And so I guess the answer to your question is resistance exercise may be much more helpful than some people expect, because they want the exercise to itself consume as much energy as possible. You may get an additional benefit in resistance exercise that you actually become a less efficient in terms of your muscle work, and that will help you to maintain your reduced body weight. So resistance exercise is probably something that should be considered. We have a paper on this. You could, I thought I had a slide from it, but you can look it up.
Great. So our next question from Li Ming, and the question is related about leptin. I think can we use leptin levels based on healthy normal weight control to guide the healthy body weight by drug or lifestyle modification for the obese individuals?
So I think, again, in our experience, and I think this is pretty generally true, there's some slight exceptions to what I'm going to say, but if you take individuals who are weight stable, either obese or lean, wherever, however they got there, the leptin will reflect the level of body fat. So what you really want to know is not the leptin concentration itself. What you really want to know is this. You want to know how that concentration, at least in the model I'm presenting, if this is the concentration here, these arrows, you can predict what this arrow will look like by just knowing how much body fat they have. There's no evidence that obese and lean people differ in the amount of leptin that they produce per unit of fat cell or fat mass, with the exception, of the male-female difference I mentioned. What you really want to know is where this arrow is relative to this. And that's something that we have not yet figured out how to measure. You're asking a very important question. The leptin itself won't tell you this, but what we really need is the ability to say how that leptin relates to this set point or threshold or whatever you want to call it, and that we don't have. If we did, we then would be able to make the kind of prediction that your question is implying. That is, where is this individual relative to his or her set point? Do I need to do something to help them to sustain this weight loss, or are there other interventions that would be more appropriate? This is an absolutely critical question, and we don't have an answer for it. If you can figure that out, I'd like to hear from you. But you asked the right question. The level itself isn't going to tell you that. It'll just tell you how much body fat they have. But what you want to know is how their body fat relates to this. And that's a question that can be only measured now by actually perturbing their body weight and see how they respond.
Okay. So the final question in the chat box is, So the question is, thanks for your fantastic talk. So nowadays they are revealing leptin sensitizers to restore the leptin sensitivity in obese ones, but the underlying mechanism still elusive. So may I know how you think about this issue? Do you agree that those leptin sensitizers are more likely to directly act on the CNS or peripheral and act on the adipose tissue?
No, the sensitizers are acting almost, well, I think the sensitizers will work by acting centrally. And what they will do is cause, if you want to think about it this way, this relationship that's schematized here, it will cause the central nervous system to hyper respond to the amount of leptin. Remember, I mentioned to you that if you give high doses of leptin to individuals exogenously, you can actually cause them to become resistant to the hormone. That may be one of the reasons why the trial that I mentioned earlier was unsuccessful, although even lean individuals failed to respond as well. But if you can increase the sensitivity of the central nervous system to the hormone itself, which is what these sensitizers are doing, you would anticipate that you would begin to see effects on energy intake and expenditure that are sort of predicted by this kind of model. So if you can do something here that enhances the signaling capacity of the system at both of these receptors, you would anticipate that you would get favorable responses with regard to energy intake and energy expenditure. And that's apparently what the sensitizers are doing. You're actually, in a sense, getting a pharmacologic response to your own endogenous hormone. It's like and you've hypersensitized or resensitized these receptors to this hormone, and you're getting additional impact of the hormone at these neurons and probably elsewhere in the central nervous system. So to the extent that they work, they would likely work in the central nervous system to have the effect that you described. And you would predict that you would be able to get at least some additional weight reduction or get some weight reduction as a result of this sensitization or hypersensitization. What the downstream effector system would do chronically to seeing this hypersensitivity, I think remains to be seen. But in theory, it should work. And it does. I mean, actually, these effects have been shown, at least in animals. But it's central. The mechanism is central.
Great. So I think that's all the questions. And so far, Yanling, do you have anything? Any questions? Or it's late, right?
Yeah, I think it's too late. I'm going to send email to Rudy, ask some question about these cerebellum circuits on the regulation of appetite. But yeah, I'm going to send email to Rudy.
Okay. I also have some question related to adipose tissue.
Okay, there's my email address. I'm happy to get your comments. Yeah, I didn't talk about the cerebellum today.
Yeah.
Okay, so thank you, and thank you, Dr. Rudy Leibel, so for this wonderful and I think very complex and research, right? And also we noticed the challenge is not only body weight loss, but actually body regain. That's the major challenge we face, and we have a lot to do for all.
Very good. Well, if I got that message across, I'm very happy. I hope you all will figure this out. And tell me how to do it. So
it's a long way.
Yeah, well, maybe not. I don't know. We'll see. You're smart.
Yeah.
Okay.
So nice to see you, Rudy. Maybe we should find another time, to chat.
Okay.
What about this year? ADA meeting? Rudy, are you gonna attend ADA meeting this year?
I might attend the ADA meeting this year. Yep.
Perfect. Okay.
Very good. Well, I hope to see you all soon.
Yes.
Sure.
Yeah. So good night.
Bye. Good
Good
night.
night. Take care. Good morning or whatever.
Yeah, it's morning here.
Yeah. Take care. Thank you.
Thank you.